Hi, my name is Catherin Shaju, and my project explores the m6A regulation of AMIGO2 transcript stability in LCC9 breast cancer cells. Breast cancer is one of the most commonly diagnosed tumors in women in the United States. Estrogen action blocking drugs are used to inhibit metastasis in estrogen receptor expressing ER-positive tumors. However, some patients develop resistance and get metastasis in other organs. Adhesion molecule with Ig-like domain 2 or AMIGO2 protein mRNA transcript has a higher number of m6As in MCF7 sensitive to tamoxifen compared to LCC9 breast cancer cells resistant to tamoxifen therapy. Reduced AMIGO2 transcript was associated with drug resistance in ER-positive breast tumors, thus bringing the question, what is the effect of METL3 knockdown on AMIGO2 protein stability in LCC9 breast cancer cells? The hypothesis was that the knockdown of METL3 will decrease the AMIGO2 protein stability. The independent variable was the METL3 knockdown. The levels of independent variable were control, SI control, and SI METL3 transfected cells. The dependent variable was the AMIGO2 protein stability. Methodology. The first step was the 8 plates of cells were set up and transfections with short interfering METL3 or SI METL3, SI control, and the control were plated. Then the cyclohexamide was incubated at the time points of 0 hours, 1/2 hour, 1 hour, 3 hours, and 6 hours, and the whole cell extract was isolated. Lastly, a Western blot analysis was completed and normalized to Ponsu and amido black stains to measure AMIGO2 protein stability. Data and results. This is the LCC9 cells blot. This is the METL3 single band fold change relative to Ponsu S. As you can see here, about 25%, there was approximately a 25% reduction in METL3 protein for SI METL3 transfected cells, thus proving that the METL3 was indeed knocked down. This is the relative AMIGO2 expression for the LCC9 breast cancer cells. In here, SI METL3 transfected cells had a significantly higher relative AMIGO2 expression overall. This is the nonlinear fits. The SI METL3 transfected cells AMIGO2 protein had a half-life less than half of that of the control. Some possible errors include cross-contamination during desonication and loading of samples into the electrophoresis apparatus. The conclusion was that the hypothesis was supported and the relative AMIGO2 expression was higher for cells transfected with SI METL3 than that of the cells transfected with SI control and the control cells. An application is that the knockdown of METL3 can be used to increase AMIGO2 protein stability in LCC9 breast cancer cells, thus decreasing the resistance to tamoxifen therapy.